இது நம்ம ஐயா கோச்சிங் சென்டர். வாங்க படிக்கலாம்.
இந்த வீடியோ உங்களுக்கு பிடிச்சிருந்தா லைக் பண்ணுங்க, ஷேர் பண்ணுங்க, சப்ஸ்கிரைப் பண்ணுங்க. Thank you.
Let us see supplementary genes. What is meant by supplementary genes? Two independent genes present on different gene loci. Each produces its own trait, that is character. When two independent genes present on different gene loci, those two genes produce its own character. But when these genes interact, when they are present in which condition? No, this is very important, dominant condition. It produces a new trait. When these genes interact, during interaction, they are present in what condition? Dominant condition. And what they produce is a new character. Next, we'll see about the characteristics of supplementary genes. First one is pair of non-allelic genes present on different loci. Second one is one allelic gene, if it is present in dominant, produces its phenotypic character independently. So, one allelic gene dominant in dominant condition produce its phenotypic character independently. But second allelic gene, when it is in dominant condition, it modifies the phenotypic effect of first non-allelic gene. See, previous point you have seen, suppose this is a first non-allelic gene. When it is present in dominant condition, they produce its own character independently. But suppose if second allelic gene is dominant, it modifies the phenotypic effect of the first non-allelic gene. At the same time, the second allelic gene may not produce its own effect independently. In which condition? Supplementary genes respectively. Now let us see the example for supplementary gene. See, in case of, this is the comb, that's contained cholvanga, the shape of comb in poultry. You can see the types. You can see different types of comb in poultry. See, the comb character is controlled by two genes. That is, each comb character is controlled by two genes. Okay, for example, the pea comb is controlled by two genes. Same like that, especially when it is in dominant condition. Then rose comb is also controlled by dominant condition. The remaining comb we'll see later. See, these two are dominant over single comb. This is what comb? Single comb. These two are dominant over single comb. Especially when they are present in recessive condition, they produce what? Single comb. Then we will see how walnut comb is formed. Let us take the example now. First parent. We can take the parent. That is first one, P. Parent which is having what type of comb? P comb, capital P, smaller. Okay, next one is, next parent, rose comb. They are having rose comb, that is small p, capital R. You can see when this present in dominant condition, it produces P comb. When this is present in dominant condition, it produces rose comb. When these two parents are crossed, first you'll form the gamete, capital P smaller, capital P smaller. These two are gamete. Same as small p, capital R, small p, capital R. These are the gametes. One crossed, capital P, small p, capital R, small r. So this is F1 progeny. Now what comb it will possess? The comb it possesses walnut. The comb it possesses walnut. Next one is, we can see in F2 generation, when the F1 progeny is selfed, capital P, small p, capital R, small r, into capital P, small p, capital R, small r. Now next we'll draw the checkerboard to find out the ratio of what types of combs are formed. When F1 progeny is selved, see I have drawn the checkerboard. Now we can find out the ratio, how many pea comb, how many rows, how many walnut, and how many single comb. We'll find out this. First one is, listen, that is capital P, capital P, capital R, capital R. Then capital P, capital P, capital R, small r. Then capital P, small p, capital R, capital R. Then capital P, small p, capital R, small r. Then capital P, capital P, capital R, small r. Capital P, small r, small r. Capital P, small p, capital R, small r. Then capital P, small p, small r, small r. Then capital P, small p, capital R, capital P, small p, that is capital R, small r. Then small p, capital R, small p, capital R, small r. Then capital P, small p, capital R, small r. Then capital P, small p, small r. Then small p, capital R, small r. Then small p, small r, small r. See, now we can find out how many walnuts. See, when two dominant are present, it produces a new trait. So it is walnut. And then next one, here also walnut. And here also walnut. Here also walnut. You can see this also walnut. And this is pea comb. Because this gene produces what comb? Pea comb. And then next you can see this is walnut. And this one is pea comb. This one is pea. Then see here, this is walnut, and this is what comb? Rose comb. And this is what? This one is also rose, because this is one dominant is here. This is also rose. And see here, this is walnut, and this one is pea comb, and this one is rose. You can see here, these are recessive condition and produces what type? Single comb. See, single comb. The single comb is produced by, in which condition? Recessive condition. And when both the dominant, the second allelic gene modifies the first one, that is the pea comb, that is rose, modifies the pea comb gene and produces a new trait. That new trait is called as what? Walnut. And the ratio is 9:9 walnut, then 3 pea comb, 3 rose, and 1 single. This is worked out by W. Batson and Punnett.